Kelambakam, Chennai. So here we have our speaker, and I think it's good to start, sir.
Good morning
Okay.
to all. Thank you, Dhanusri, for your brief introduction about myself. Am I audible to everyone?
Yes, sir, you're audible.
Is the screen is visible?
Yes, sir, it's visible.
Okay. So before I can go to this marine pharmacology, I want to give you this brief outline in the marine. So what is marine? What is the species which is present in the marine organisms? Then accordingly, we may going to see these types of species, how it is going to be taxonomically characterized from the marine. Once it is taxonomically characterized, how it is taken for the biomedical application as well as biological application. So coming to this introduction, marine has huge floral and faunal biodiversity, estimated approximately to be over 5 lakhs of species when compared to terrestrial species. So many of the species which is exist in ocean, which is not in the cases of terrestrial. In addition, marine organisms are rich sources of structurally diverse, unique chemical structures and potential bioactive compounds with varied biomedical activities. These biomedical activities, which is include antimicrobial, antiviral, anti-cancer, and then other activities of these biological applications, which is going to be included in this biomedical field. Marine mealy serves as a resource for potential functional material, which include in the name of the biomaterial, which may be fall in the category of polysaccharides, or maybe an peptide, or maybe an polysaturated fatty acid. In addition, it may exist a predominant diversity of mineral content, vitamins, and all these bioactive molecules, which plays a vital role in the form of antioxidant, and sometimes it is acting as an enzyme also. So coming to this drug discovery, so current trend in drug discovery is to understand how the infections or the disease are controlled, which is based on the molecular pathway and physiological levels of the drug candidate, how much amount of the knowledges which can be having in the form of infections, as well as the management of this drug inducing things, which is going to be suppress this causative factor. There are about numerous advanced technology and emerging knowledges are still yet to be analyzed in the field of biological system. Still, the drug discovery process is under juvenile state only because of this slow emerging system, and this leads to causes this new therapeutic discoveries. With this limitations of this drug discovery, the ocean is also plays a vital role to identify the drug candidate. Once you are identified this drug candidate, which leads to make it this isolation process and then structural characterization. So once you are comparing this completing this isolation process as well as structural characterization process, after that it is taken for screening of biomedical activities of the purified product which is obtained from the marine species, maybe it is in the flora or in the fauna. And after this drug efficacy, which is done in both in vitro as well as in vivo, and it is taken for this finding this suitable drug candidate, and based on this drug candidate output from this experimental model, and it can be taken to this clinical trial. So there are few drugs which is taken to clinical trial 3 stages from the ocean. And coming to this first species which we are going to take it for this marine pharmacology field of update in Chettinad, we are looking mostly the drug like compound or the drug compound from the seaweed as well as mollusk. So this is the 2 things which we are routinely concentrating in Chettinad. So before I can go to this giving this materials which we obtained from these seaweeds, I want to give you this a brief outline of this seaweed. What is seaweed? So seaweed is nothing but it's a macroalgae which is classified into 3 categories. Maybe it is depends upon the phenotypic characters. It can be classified in the form of blue algae, green algae, red algae. So, and the species which is shown in the images here, which is in brown algae as well as green algae. So with this species which is obtained from this Gulf of Mannar, where this national biopark which is located in this Rameswaram district, and it has a high salt tolerable capacity, and the seaweeds which has a enormous amount of minerals as well as vitamins. In addition to this, which is a suitable species for the agar extraction. So most of the agar which is obtained from the chemical industry, this is the only sources which can provide this agar. So based on this yield, we are also collecting this species from the Gulf of Mannar, because this Gulf of Mannar is a huge biodiversity of these seaweeds as well as sea grasses. So the seaweeds, once which is isolated from this Gulf of Mannar, which is brought to this laboratory, and it should be cleaned, the salt and then sand and other debris, which is thoroughly cleaned with the tap water followed by the distilled water. After that, it is taken for the shadow dried. Once this shadow dried things which is taken for further processing of isolations of the phytochemical from the seaweed. So, and the picture which is showing here, this is the cleaned seaweeds of Laminaria digitata, and this is the picture which is showing this field photos which we collected from the Gulf of Mannar. So the once this seaweeds, if it is all the debris, if it is removed from this seaweeds, it is taken for this shadow dried. Once it is shadow dried, it is taken to this blender, and with the help of this blender, it is powdered, and the powdered things, it is taken to this aqueous extract. Aqueous extract we used to follow hot water as well as cold water. So the hot water extract, which can take it this all the soluble form of carbohydrate which is exists in the agar, it is going to be soluble, and it is polymerized. So this picture which is showing the agar which we are extracted. So once the agar which is extracted, it is taken for this structural characterization as well as biomedical application. So one of my student interested to test this agar as an antioxidant as well as anti-aging product. So for this, she selected D-galactose is an aging-induced chemical. Why we are taken this D-galactose is an aging-induced factor now? Because this D-galactose is a readily available carbohydrate which is less expensive when compared to other aging-inducing factor. So whenever you are going for aging factor, you need to identify these physiochemical parameters in addition to this biochemical as well as genetic factor. So we have obtained this animal ethical clearance from this Chettinad Hospital and Research Institute. The Swiss albino mice, female mice, which we are selected for this study. So this slide shows these behavioral parameters which is confirming this aging induced as well as control group, and followed by the treatment group. And first two photos which is representing this photometer as well as a light dark exposure, and this is the heart rate, and this is the rotor heart. So and we are going to take
excuse me, sir.
Yes, sir.
Sir, your slides are not changing.
Are you able to see the slide, sir?
No, sir, it's still this first slide, sir. Could you please reshare?
Wait a minute. Now it is able to see the screen.
Yes, sir. Yes, sir. Control the galactose.
Yeah,
yes.
yeah. Just see these behavioral parameters which we are confirming by these four factor. One we are taken for photo octometer, and another one is light dark explorator, then heart plate assessment and rotor heart. So this is the four factor which is taken for confirming this aging induced factor. So the total durations of this experiment which we are taken for aging induced galactose, it is up to 49 days. So the second week of D-galactose, which is subcutaneous induced to this Swiss albino mice, which shows this aging pattern, which is clearly shown in the abdomen part of this mice. Just to see this, when compared to control, this D-galactose induced mice which shows this skin shining in the abdomen, whereas this third photos as well as fourth photos which is showing the the sample treated agar treated followed by the positive control which we are taken for the vitamin C. So, and this picture which is representing this histopathological confirmation, we have done this biochemical parameters followed by these histochemical parameters, and these histological evaluations of the skin patterns of the control which does not shows any fat bodies depression when compared to this induced thing. So this figure B, which is representing the fat body deposition, which is clearly indicated in the arrow mark, whereas the agar treated as well as vitamin C treated, which is minimizing this fat deposition. And from this we come to conclusion, the agar which is extracted from this lamellar digitata, which has evidence based proof of this aging postponing things. So, and one more student, and she is also concentrated this study for her final semester project work, and instead of aging she wants to go for anti-cancer activities. She also interested in the agar, the same species which we selected. The seaweed here also we have selected for this laminar digitata. What is the difference between the agar which she taken for the previous studies and this present studies? Whereas the previous studies agar, it is taken as such as a raw material and it is given to the mice, whereas this girl selected the same phytochemical in the form of agar where it was formulated. So after formulation it was taken to topical application, so topical external application. And here see in the taken this dimethyl benzyl anthracene is an skin cancer inducing chemical. So, and after getting this ethical clearance from this Chettinad Hospital Research Institute, and we have taken this formulated agar, and this picture which is showing the stability of the formulated agar which we are done in our laboratory. So up to 3 months the formulated agar does not shows any microbial content and does not shows any changes and odor and homogeneity and consistency and pH. All these are all the various pharmaceutical parameters which we are tested from the month 1 to month 3. Up to 3 months it does not shows any morphological as well as physiochemical characterization changes. In addition to this, we have taken for this structure analysis patterns also. If we are seeing this moisture content of this formulated agar, which is account for 48.28%, hardness and adhesiveness I have given here in the tabular column, followed by the elemental and then mineral analysis also we have tested by using this atomic absorption spectroscopy. And just to see this sodium, potassium, calcium, and then phosphorus, magnesium, all these are all the various elements which is present maximum amount of the species which we are selected. And after this dimethyl benzyl anthracene which we are applied in the group 1 and group 2, sorry, group 1 acting as an control, whereas group 2 and group 3, group 4 and group 5 which is acted as a cancer induced by using DMBA. And just to see this group 2 photos which is shows the melanoma which is size account for this 75 millimeter in size, and the another pictures also which is showing this 47. Well, but when you are comparing this group 3, the sample treated, the formulated agar topically treated mice which reduces the cancer size, which is account for 39, and the same thing for 2 doses we have selected. One is low doses, another one is high doses, and the low doses which is reduces size of the cancer up to 39, whereas the high doses which is reduces the cancer up to 32 millimeter, whereas the positive controls also which is accounts for 43 millimeter. And after this experimental period, the mice was taken for this euthanistic condition by providing this vitamin. It was sacrificed. Skin parameter and blood parameters it was taken for confirming the anti-cancer activities of this formulated agar treated and then DMBA induced things. So P53 levels And addition to this P53, we have taken for keratinocyte content estimations also. And this P53, the control, when compared to control, the DMBA induced cancer, which is significantly increased, this was significantly reduced in the group 3 and group 4 and followed by the positive control. And the same type of expressions we obtained for this keratin content also. The keratin content which is less in the cases of DMBA induced skin cancer mice, and the keratinocyte which was significantly increased in the low dose and then high dose when compared to this positive control. So from this results, we come to conclusion the formulated agar which we are taken for the mice anti-cancer property, it has a good effect of preventing the mechanism, which was confirmed in this histopathological analysis also. And just to see these skin patterns of this histopathological analysis of control, and A which is indicating the control, whereas B which is indicating this skin patches which was widely denatured in the skins of this DMBA induced things, which was minimized in the cases of low dose treated formulated agar as well as high dose treated of formulated agar, which were compared with the vitamin D treated mice also. So from this also we come to conclusion. So the formulated agar which we are obtained from the laminaria digitata, it has an anti-cancer properties against this DMBA induced skin cancer. So the next one, so far we have discussed this phytochemical compound which we are tested in the laboratory. Now we are going to this animal sources. What are the types of animal sources which is going to be provided for this drug? And so we have taken this mollusk is one of the routine animal which we are taking for this testing this bioactive compound from this mollusk, and this compounds also we have taken anti-cancer activities as well as neurological aspect. So before I can go to this result, I want to give you this brief outline about this, how these mollusks are plays a vital role in the field of nutraceutical as well as in the biomedical applications. So there are about 2 lakhs of species which is accounts in the classifications of the mollusks. So mollusks are soft body animals. Mostly it is call it as a snail or the oysters. Sometimes the higher categories which is bring it in the conditions of octopus as well as squid. So generally the mollusks are classified into 7 categories, but major classifications of the mollusk, if we are taking now, Bivalvia, Gastropods and Cephalopods. So I will give you this difference between these 3. So just to see this uses of mollusk to the human being, which is acting as a food, and it gives this fuel, both ornamental as well as nutraceutical aspect, which plays a vital role. So since 200 years and ago, so people they are taking this mollusk meat as a food. And the first picture which is showing this bivalvia, so it has 2 cells which is connected with a single wall. So the example for this bivalvia, you can take it clams, oysters, scallop and shrimp worms. So this is the subtypes of this bivalvia classifications. So in this bivalvia, it doesn't contains any head or the radula. So why we are saying here head and radula na? If you are going for gastropod as well as cephalopod, so I will show you the cephalopod next to this slide. So the shell which is spiral or it can be in conus. So this is we can bring it under the category of this gastropod. The gastropod as well as cephalopod which has a toxic substances, and these toxic substances which plays a vital role in the field of cancer biology as well as molecular biology. So the toxins which is present in this gastropod as well as cephalopods. So the cephalopods picture which I am showing here, you might be seeing this locally we can say it as an convoymine or the cuttlefishes. So this is the octopus and this is the squid and this is the nautiluses species which is end-ended, and this is an space we can say it as an Cephalopods. So all these are all the photographic representation of this classes of cephalopod. Why this is called as a cephalopod? The head and the foot which is together in the species we can call it as a cephalopod. So from this mollusk, which has a heteropolysaccharide, especially in the form of anticoagulant like heparin and heparin-like substances, which plays a major sources in the cases of this cephalopod. So in Chettinad, we are looking for this heparan sulfate isolations from this mollusk also. Why we are going for this heparan sulfate? No? Because you know the clinical applications of this heparin, which is an anticoagulant from 300 years ago, but still the commercial sources which you are concentrating for getting this heparin based material, maybe you can obtain it from bovine sources or the porcine sources. So the bovine sources and porcine sources, if there is any viral infection, if it is present in the sources now, which leads to infect the patients also, if we are taking these infected sources as a heparin isolation. And moreover, these bovine sources as well as porcine sources of heparin, which has an religious restrictions of usages of the pharmacology aspect also. So with this, we are concentrated this non-mammalian sources in the form of marine mollusk we have taken, and this animal which we are taken for isolations of this heparan sulfate, which is represented here. The scientific name we can call it as an Amusium fluoronectus. In local terminology, if we are taking this confirming this species, we can say it is moon cell or the sun cell. So this is a dorsal view and then ventral view of this, and this is the opening of this bivalve. So after this animals which were obtained from this ocean, it is brought to this laboratory. All the soft bodies which is present in this mollusk, which was removed from this shell, and it is taken for further hydrolysis of the lead molecules identifications in the form of heparan sulfate. So this is the structures of the heparin, which is made up of Iduronic acid as well as glucosamine. So just to see the flow chart of this isolations of heparan sulfate. So if you are taking marine sources, it is taken to this homogenation. You need to remove this fat. After that, you need to take it for the proteolysis, followed by this proteolysis. You can take it for chemical hydrolysis of proteolysis as well as enzymatic hydrolysis of proteolysis. Also, it is applicable for removing this fat as well as protein from the selected sources. What is the advantages of going for taking this proteolysis by using this enzyme? Now, it is gives maximum amount of the yield, as well as when you are going for structural characterization, enzymatic hydrolysis process, it gives an NMR spectrum properly, whereas the chemical hydrolysis of the heparan sulfate, it has the trace impurities of chemical integrity, so it is very difficult to get it. From my experience, I am sharing these things. After removing this fat content and then protein content, it is taken for three types of chromatography: ion exchange as well as gel filtration chromatography. So similar to peptide as well as protein, we don't have a commercial suitable column for this heteropolysaccharide, especially in the cases of heparin. So that's why we supposed to take it more than one nature of the chromatography to get the single nature of the glycosaminoglycan. So we have taken this chromatography amber light, followed by this DAE cellulose. After that, it was taken for Cephalopods G100 column chromatography. All these three chromatography, which is identified this active fraction, and after that, it was taken for the freeze drying, and followed by the freeze drying, it is taken for both structural characterization as well as biomedical application. So, and this picture which is showing here, which is isolated from the marine sources of NMR spectrum of this heparan sulfate, whereas this is the standard. You just see this chemical shift of this standard as well as marine mollusk, which shows a similar pattern. So from this also we can come to conclusion the technique which we followed in the laboratory, which has a maximum purity. So I have given here labeling pattern A, B, C, and the same thing I have given the standard also. So just to see the tables pattern. So what is H1 and glucosamine and all these things I have given here. The standard glucosamine pattern, which is observed around 5.61, whereas in the isolated mollusk, which shows 5.41, and glucosamine, which is obtained in the chemical shift of 5.15, whereas in the heparan sulfate from marine mollusk, which is 5.26, and followed by this Iduronic acid and glucosamine and methyl groups, I have compared the previous NMR spectrum with this tabular column clearly. After this confirming this NMR spectrum, we have taken for other element analysis like carbon, hydrogen, and sulfate content, how much amount it is present in the cases. So this was shown in this bar diagram. So the blue color which is representing this standard heparan sulfate, whereas this orange color which is representing the elements concentration in the isolated marine molluscan samples. So almost the heparin sulfate which is obtained from this marine, which has an equal element content of standard. And after this confirmation, we have taken for this agarose gel electrophoresis pattern confirmation, followed by polyacrylamide gel electrophoresis patterns also. Why we are taking these two electrophoresis for confirming this heparin? So you know, these heparins are classified into three categories: heparin 1 and heparin 2 and heparin 3. So what is these three now? Based on the molecular weight, it is classified into high molecular weight heparin, medium molecular weight heparin, and then low molecular weight heparin. So low molecular weight heparin, which is routinely used from the clinical sector, heparan sulfate. So heparan sulfate, which is treated with heparin lyase 1 and lyase 2 and lyase 3. So whereas heparin, it is hydrolyzed by all these three enzymes, whereas heparan sulfate, which is hydrolyzed only by this heparin lyase 1, which is not hydrolyzed by heparin lyase 3. So which is given here. So L1 represents this lane 1, which is the standard, and L2, which is the isolated heparin treated, and then bovine heparan sulfate treated with this heparin lyase, which is shown in the lane 3 and lane 4, which is the isolated thing. Whereas heparin lyase treated, this is the standard and this is the isolated thing. So from this we come to confirmation the isolated glycosaminoglycan from the marine mollusk, which is not an heparin, which is an heparan sulfate. So all these are all the physicochemical characterization. We have confirming these things, and after that it was taken for molecular weight determination. So the molecular weight determination, why we are concentrating now, I told you, if you have this molecular weight less than 500 or around 500, now it is easy to confirm your biological assay if you are taking for a rodent model or if you are taking for the clinical sector. So the molecular weight of this isolated marine mollusk heparan sulfate, which is account for 690 Dalton. So this is not yet reported earlier, as well as this what is this least molecular weight of heparan sulfate, which is not applicable in the cases of porcine as well as bovine. So I told you when I come to the discussions part of this heparan sulfate clinical usages, if the molecular weight of the heparan sulfate, if it is maximum now, which has high anticoagulant activities, if it is low now, which has less anticoagulant activities, this is depends upon the sulfate position of the heparan sulfate. Maybe the sulfate position, it can be in the 4th carbon atom or it may be in the 6th carbon atom. The position also which plays a biological application, and then the molecular weight also, it's also plays a vital role. So if you have least molecular weight of heparan sulfate or any anticoagulant, it is not needed to give antidote of this heparin, like histamine like substances. Histamine is an antidote for the heparin. So histamine, which is an hemophilic agent, whereas heparin anticoagulant. So and this is the advantages of going for the selecting least molecular weight heparin. So in the lab, we have tested this cardioprotective effect of this low molecular weight heparin by inducing isoprotonol is an damaging agent in the heart. So what is the mechanisms of isoprotonol, which can generate this reactive oxygen species, and it leads to causes the single cell defect in the myocardium. So after getting this animal ethical clearance, we have taken these 4 groups of rat, Vista rats, it's taken for these studies. And the group 1, which is acting as a control, group 2, which is treated with this only the heparan sulfate, the concentration I have given here, and then group 3, isoprotonol treated only 2 days, and that is also subcutaneous. The dosage is 85 milligram per body weight of the kg. Then group 4, pre-treated marine mollusk heparan sulfate followed by isoprotonol treated. So this is the experimental group setup, and the durations of the experimental was up to 2 weeks. So after that, it was taken for the biomarker assay, So the picture which is showing this ischemia, which is developed in the myocardium. This is a normal heart, whereas if you have this inflammation, or it may be in a group of cells, now we can call it as a myocardial nephropathy. If it is only single cell, now we can call it as a myocardial infarction. So the thiobarbituric acid reactive species, which we are taken from the plasma as well as the heart. So the group 3, which was maximum in the cases of plasma T-bars, which was minimum in the cases of pre-treated and then treated samples. And the same cases, the T-bars in the heart tissue, which was significantly less when compared to control as well as the treated. So from this we can come to conclusion, the heparan sulfate, which has an cardioprotective mechanisms against these isoprotonol induced things. So after that, we have measured this heart weight of rat also. Group 1, which is giving this heart weight up to 550 milligram, and the same cases for the molluscan treated rat, whereas in the group 3 isoprotonol, which is account for nearly 850 milligrams heart weight was obtained by the isoprotonol treated, which was significantly minimized in the pre-treated molluscan heparan sulfate. What is the reason for the heart weight which is increasing in the experimental mice? Because of the heart myocardial infraction, which leads to get it ROS production, that ROS production which makes the enlargement of the myocardium in the tissues, which is clearly going to be shown in the histopathology. I will show you the results later. Before we can go for this histopathology, the marker enzymes like creatine kinase, lactate dehydrogenase, aspartate transaminase, and alanine transaminase, both serum as well as heart tissue, which we are taken. Just to see these red color highlighted things. Group 3, which shows significant reductions of this marker enzyme, which was increased in the pre-treated molluscan samples. This was compared with this control as well as molluscan samples alone treated. So from this, we can come to conclusion the heparan sulfate has an cardioprotective mechanisms also. And we come to isolations of the isoenzyme pattern, lactate dehydrogenase, which is an enzyme confirming this myocardium functioning properly. So usually the LDH4, it is not going to be exist in the serums of these mice, and this was clearly shown here, whereas the isoprotonol, which shows all these isoenzyme subdivision like LDH1, LDH2, and LDH3, and LDH4, and LDH5. So the isoprotonol treated and then heparan sulfate treated, which does not produce this LDH4 expression. So from this we can come to conclusion the treated sample has a cardiac protective mechanism. So which was taken for this densitometric scanning by using this serum heart tissues which we are taken, and just to see this level of expressions of the densitometric gram. So from this we can come to conclusion. Apart from this, we have taken for this uronic content as well as glucosamine content, so which is account for 51% and then 38% respectively. And after that, we have taken for this anticoagulant activity. So the anticoagulant activities, we have taken only 2 parameter: activated partial thromboplastin time, which is shortly represented APTT, and the marine mollusk, which shows 135 international unit per milligram, whereas the prothrombin time, which shows 100 international unit per milligram. This was compared with this standard, which is readily available in the market. So which account for 170 in the cases of APTT, whereas in the PT, which is account for 125. So from this, the extracted heparan sulfate from the marine mollusk is also an alternative suitable substances for the anticoagulant. So next to this isoenzyme pattern, we have taken for this ECG patterns of this rat, and just to see these ECG patterns of the rat, where the PQRST elevation, which was normal in the cases of control rat, and the same thing for the molluscan heparan sulfate treated rat also, whereas the C, which is indicating the ST segment, which was maximum in the cases of isoprotonol alone treated, and this gives these cardiotoxicities. This was minimized in the pre-treated heparan sulfate, and from this also we come to conclusion. And final confirmation which was taken for the histopathological analysis also. So the slide which is showing here, which shows the normal myocardium pattern, whereas the myocardium edema and swelling which was observed in the cases of group 3, which we are taken as an isoprotanol, whereas this B, which is showing the molluscan samples alone, whereas the group D, which is showing the edema and the swelling of this myocardium, which was minimized by the molluscan sample treated rats. So next, polysaccharide, which we are taken from this Chettinad is chitosan, which is also a natural polymer next to cellulose. Chitosan, which is a natural polymer, which is account for various biological activities. The commercial sources, which is routinely used for confirming getting this chitosan, either it can be obtained from the crab shell or the shrimp shell. So what is the limitations of these commercial sources now? White rot fungus or white rot virus diseases infection, which is quite common in these two species. So we need to have an alternative sources. So we have taken this cuttlefish. So if you are taking cuttlefish now, you may going to have the cuttle bone, which is a non-edible. So it is going to be discarded as a waste. And this is the squid, and this has a gladius, and this is also a non-edible. It is discarded as a waste. So we are taken this waste as a resources for a drug candidate. So one of my PhD student, he wants to check the neuroprotective effect of this low molecular weight chitosan against this pesticide induced toxicity in the zebrafish model. So this slide shows this sulfated chitosan and then other derivatives of chitosan, how it is going to be used. The chitosan, it can be taken to sulfated chitosan, or else it can be taken to carboxymethyl chitosan. Either it can be taken for nanoparticles of chitosans also. Why we are concentrating particularly the chitosan? What is the limitations of chitin? Chitin is the second most large polymer in the earth. It is highly insoluble in neutral pH. So if we are converted to sulfated chitosan or carboxymethyl chitosan or acylated, it is soluble in the neutral pH. So that's the purpose we are converting chitin to chitosan and chitosan to other derivatives. So based on this low molecular weight conformation, again we have taken for this MALDI the molecular weight of this sulfated chitosan, which we are obtained from this cuttle bone, which is account for 600 Dalton. Due to lack of time, I am not showing the images. Just see the occurred in orange staining patterns of this zebrafish brain sections and the larva. The control, it does not show any neurodegeneration, whereas in the rotenone treated, which shows this neurodegeneration, which is minimized in the cases of positive control. Levodopa we have taken, as well as the carboxymethyl chitosan and sulfated chitosan treated, which is also minimizing the things. So from this, we can come to conclusion: the carboxymethyl chitosan as well as sulfated chitosan, low molecular weight, which has a neuroprotective effect, which is induced by rotenone. So rotenone is a natural pesticide, so which was confirmed by this histopathology also, and we have taken these sections of this brains of zebrafish, and it is taken to eosin and then emission staining, and the magnification which we are took for this is 40x. So the control which does not show this neuronal disarray, whereas this rotenone treated, which shows this neuronal loss of very decreased cell mass in the brain, which was minimized in the cases of low molecular weight sulfated chitosan and rotenone treated, and then low molecular weight low dose and high dose. So which is proves this low molecular weight sulfated chitosan as well as carboxymethyl chitosan, which has a protective mechanisms against this rotenone induced toxicities. So far we have seen only this carbohydrate, and now we are going to move to some of the toxic substances which is also plays a vital role in the field of pharmacology. So what are the toxic substances which is going to be routinely present in the marine species? Now you can take it, the picture which is represented here, you can take it sea snake, and you may going to have the cone snail, and then lionfish. All these are all the highly toxic substances which can be used for its prey, and it was compared with the terrestrial venomous species also. So, and some of the picture representation of this toxin which is obtained from this conus, and this conus also classified into various subtypes. But as a life scientist, if we are taking this identification, you need to have in the form of whether the conus it has an piscivorous or molluscivorous or vermivorous. So the feed of conus itself, it can be classified into this subcategory. So the this is the examples of this conus, and this is the feed of the this conus. So this slide, which shows the cross sections of the venom or the toxic substances, just to see this is a venom bulb. Whenever this conus, which is a sluggish animal, so whenever if it wants to get feed for its systems, it can release this venom, and this venom which is going to be released to this radula, and this is a photographic images of this radula. So this radula, this is also a radula. So just see this. This is a conus snail. When the small fishes, maybe the clown fishes, if it is come closer, it can release the radula, and this radula, which is already connected this venom, and it is going to be paralyze this species of this clown fishes. After that, it can be engulfed by this parapaces of this conus, or else it can be has like a making a fish net. So like these patterns also, it is going to be used its toxin for its capturing the prey. So this is the natural mechanisms of this toxin from the conus. And what are the applications of this conus? As literature supported us, this conus which has a toxic substances, and the toxic substances which has a neuromuscular disorder, it is going to be treated, and which is going to be used for anti-convulsion and anesthetic drug also. Sometimes it is used for painkiller. I told you some of the conus toxic substances, it is under clinical trial 3. So it is used for painkillers also. The alternative drug which is readily available, morphine. So and zincotic. So this is a one of the toxic substances which is obtained from the conus, and this is still under clinical trial 3. Maybe we can expect this zincotic in market after few years later, which is used for chronic painkiller. So with this, we come to this end of the session. So I want to give my sincere thanks to my research team. Just to see this picture, which is giving this my research teams, and 2 PhD students who made enormous contribution in the native medicine and marine pharmacology laboratory in Chettinad, and he is Dr. Karthik, and he is Dr. Manikandan. So these 2 guys completed their PhD under my guidance. Now they are working as a postdoctoral fellowship at Texas University, USA, and this is the research team, and this is the 2 PhD scholar currently doing their PhD under my guidance, and this is the MS and PhD project students. So now we move on to this acknowledgement. My sincere thanks goes to Department of Biotechnology, Government of India for providing this extramural funded project. In addition, my whole heart thanks to Dr. M.V. Ramanamurthy, Scientist G and Director, and then Dr. T. Sanmugara, Scientist Chef from National Center for Coastal Research, which is a sub-autonomous body of Ministry of Health Sciences, who has given permission to utilize the facilities of Gulf of Mannar National Park the last year. So with their without their support, I cannot went for the field work. I can able to lead to make it some of this phytochemical compound from the seaweed. This aspect I want to pass my sincere thanks to this both the scientists from NCCR. So guys, if you have any questions, you please unmute and ask me.
Thank you, sir.
Thank you, sir.
So we have a few questions in the chat.
Yes, sir.
Questions from Babu.
So do we have marine drugs for COVID? So still it is not under the category of COVID. Maybe in future, because the COVID it came 2 years before. Some of the review of literature I can able to share with you, but it is not a single nature of things. Maybe in future you may expect this drugs from marine, which leads to cure the COVID also. Next question, what is more likely to responsible for bioactivity in cancer treatment, ceramide or glycolipids? So it's not a ceramide, which is an agar, which is making this skin pigment in the form of keratinocyte or the connective tissue protein like collagen, as well as one more protein, myosin. So that two protein, which is going to be enhanced by this agar if we are taking as an topical application. So that's a mechanisms we understood from this publication. And one more question: how counter current chromatography used for extractions of these marine drugs? So I told you, sir, counter current chromatography, which is mostly used for peptide based or toxic substances if we are targeting. No? If you are not interested in the aspect of peptide, other than peptide and then toxic substances, suppose if you want to go for fatty acid, or if you are going for carbohydrate, no? This counter current chromatography, which is not going to be apt for this purification of that two molecules, except the protein or the toxic substances. Hello.
Okay, I think he got the answer, sir.
Okay.
Um, so, like, I have a general question.
Yes,
So,
ma'am.
regarding all the research that you have done regarding the marine pharmacology, so what is the expected outcome that you're trying to give to the mankind? Like, is it the development of drugs, or how is it, sir?
It's a valid question. I can say some of the invention in my lab still under pipelines for the patterning. Once it is confirmed this patent, whether it is granted or it is not granted, I can say if it is granted, this is the drugs which we are standardized from the laboratory. I have shown only the few things. Many of the things which is not shared here because of this patterning issues. If I shared this anything to this public, it cannot be taken for the patterning. Two more patterning, it's still under pipeline. Maybe I told you these things now. The DVT also supported me for antiviral activities. So once I got the grant, I can say this molecule from that species which has an antiviral compound. So still we are not come to conclusion. It's still under pipeline only. It takes another few years.
Okay, sir.
Any more doubt from audience side?
Sir, also, yeah. So, more thing that you spoke about the rats and all, yes. But then regarding the omega-3 fatty acids that is basically seen in the fishes, have you made any study on those? Like, eventually, if we keep extracting omega-3 fatty acids from them,
My
they will
voice,
go extincted, right? So, is there any alternative for that for now?
your voice is breaking, Ma. Can you repeat one more time? The what is the questions?
So, is it audible now?
Yeah, yeah.
So basically talking about the omega fatty acids, so we get from the fishes. So if we keep stacking the omega
Again,
fatty
it has an audio
acids from the
issues.
fishes.
Maybe I can tell you the answer. Maybe I can assume what you are asking. Whether there is any specific fatty acid which is obtained from the fishes. Am I correct? Hello. Am I audible?
Yes, sir.
The fatty acid, why we are not concentrating?
I
Because
was talking about the omega-3 fatty acids specifically.
that's what, ma. Omega-3 fatty acid, there are commercially available tablets are there in the pharmacy, which is taken from cod liver, fishes, and it is already there in the market. And moreover, people who is having the habit of vegetarian only, they are going to concentrate for taking that kind of tablet. If they are being in non-vegetarian, it is not necessary to take it that tablet for their what is that nutraceutical aspect. So I think if they are in non-vegetarian, they can take it that edible fishes, the edible fishes which has enormous quantity of omega-3 and omega-6 fatty acid. So that's what one or two industry, apart from this one or two industry, they are not concentrating for fatty acid.
Thank you, sir. So she's facing some internal issues. Anyways, thank you so much, sir, for the session today. Usually we hear mostly about land animals being used, but it's the first time actually I'm being introduced to this section. So thank you so much.
Thank you, sir.
Thank you everyone for joining, and thank you everyone.
So before we can end the session, I want to pass my sincere thanks to the organizer, like Dhanushri and their team, who has given this opportunity to interact with this through online. Thank you.
Sir. Thank you for coming.
Thank you, sir. Thank you for accepting the invitation. Thank you all for listening to